That's fine. Okay, so hello everyone. We would in today's tutorial we would like to focus on zonal statistics and QGIS, and in particular we will understand what stroke plots are and how to run zonal statistics in QGIS, and finally how to compare and interpret our treatment difference among all of the strips that we're gonna create. So first, what is a strip plot? It could be any particular geometric shape or treatment zones that is laid out across our field is called the strip plots. For example, the first figure, the first plots are not considered as a strip treatments because they are only covering parts of the field. And generally speaking, the strip plots or strip treatments are running from one side of the field to the end of the side of the field. So you can see this here we have 6 strips. 1, 2, 3, 4, 5, 6. This is called strip plots. So now let's just move to our practical sessions, which is this is more practical. Let me just close my okay, so QGIS and open again my QGIS. So you open your QGIS. Oh, I haven't shared. No. Yep, this is the screenshot, I guess. Um, yep. So cool. Now, just here, once you open your QGIS, here will be a number of projects that you have worked in the past that will be displayed here. But for this, we will create a new project. You double click on the new project, and here we will try to run our zonal statistics. So first of all, we need a base layer for running our statistics. What we're going to do, we add a raster layer. So you go to the layer and then raster, you hit browse for example, uh, browse your folder. Here I have in my desktop, I have created a student folder. Here is a mosaic map of this ACLOT, ACLOT 2 UNSQ, which is located near the P9. So this is the NDVI map of that ACLOT 2, which I created for my field. And this was a drone map, imagery collected, and we have created auto mosaic, and then I have calculated the NDVI map or created the NDVI map. Then you hit open and add your imagery here. And so this is in black and white. First, if you open in QGIS, then you're gonna do like double click on that if you want to give it a nice color. Here in terms of the render type, or you can go to symbology. Maybe in your case it will open this page. You just go to symbology and try to come to the render type and select here single band pseudo color. If you hit this, select this render type and then hit apply and OK. Now you can see this is a nice NDVI map for our plot 2. So our like P9 is located somewhere here. So here you can see, let me give you a bit of information about this trial. This is my research trial, which I have for my PhD. So you can see here is these are wheat crops that we have planted on June. And we have 1, 2, 3, 4, 5, 6 strips that we have. And these are the grass that are around around of our experimental field that are grown covered by grass. So here is the bare soil, no crops is in here, and these are the wheat rows that you can see easily. So what we're gonna do is we try to create strips over this field, and we want to try to make a composition or a combination of six or three different strips or vegetation in city, for example. One we will create for the grass, and one half grass and a half soil. So, and then we will try to create three, like 1, 2, 3, 4, 5, 6. Then we will create each, we will consider two of these strips as a one for this workshop. And let me give you the length of this field from this, for example, from here up to here is 134 meter. So then the weight of each like strips from here to here is 2 meters. Since we will be considering two of the strips, then it will be 4. So now let's create our strips. What we can do is just come to our vector, click your vector, and here you have a research tools, and then on the first option you have a create grid. Then once you click there, this page will pops up. Here you have grid type, selection. There is a bunch of options that you have, and you can use whenever that suits your project. So you can go select either point grids, line grid, or rectangular, like diamond, hexagon. So any types that you like. For this workshop, we'll go rectangular because it's going to be rectangular, like 4 by 134. So, and the extent of our grid will be our field. There is also a bunch of options that you can either select the layer that you have, but for the convenience of this project, we select draw on map canvas. So once you selected that one, you just select the drag, and it will automatically consider your grid extent. And then you have the horizontal, like uh, spacing or the width of your horizontal strips that you want to consider. As I said, I will consider 2 by 1. As I said, this is 2 meter wide. This is also 2 meter wide. We will consider 2. It's going to be 4 meter. So 4 meter, and make sure for example, the unit is meter, because you have a bunch of options. You can select whatever you like. So the vertical spacing, as I said, the length is from here up to here is 134. So we're gonna select as 134. So mirror, and then you do not touch any of the other like um, overlay or other options. You do not change the CRC, for example. Make sure your CRC is 32 756, because if you select other CRC, those are not in the units, are not in mirror. Those some of the units or some of the CRC are only in grades. So make sure to select the right CRC, and then here you can save your project. If you right click on just click on these three dots. Here you can create a temporary layer, save to file. So if you want to save this as a permanent layer, you can hit the save to file. But for this workshop, we don't need to save it as a permanent layer, or will save it as a temporary layer, and I do not touch this box. I just hit the run button, and it will create our strips. So as I said, we will create like a nice mix of vegetation, semi-vegetation, 3 replication of our wheat, and also 1 bare soil strips. So it means we do not need this additional thing. First of all, you double click on the grids and come to the symbology and click the, okay, so let me show you. Yeah, symbology labels, you click the symbology and come to the simple field. And here you make it fully transparent. You just need to make it to the 0 and hit the apply and okay. So now you can see how our field looks like. And since we do not need some of these like strips, we only want to create 6 strips. So the additional strips can be deleted by just right click on this layer and try to turn this toggle editing on. You click on that, now you're able, so this option pops up, now you're able to do the removing things. Then you can, for example, so you can come to the select option, select by feature. You can either select by polygon, freehand, radius, sorry. And for our case, we only select by feature. So what we're going to do is just drag and select. Once it's selected, you hit the delete button, the delete button in your keyboard, and it will be deleted. So also we do not need all of them. We only need 6 strips. 1, 2, 3, 4, 5, 6. So then we select the rest and get rid of them. Okay. Now we have created 6 grids, strips. 1, 2, 3, 4, 5, 6. So, but we need to adjust it nicely according to our field here. So how we can do that is just select all the polygons or strips. Then here we have a bunch of tools that you can use. Here is 2 options. For example, you can move the feature by selecting this feature, or you can just copy and move that copied feature only, not the main one. Here, for example, using this, tools, you can rotate your polygons, and here you can expand it. There are a bunch of tools you can go through and try each one of them. So now we want to try to rotate our strips just to place it nicely on our experimental field. What we're going to do is just select by feature and drag it. And once selected, we try to rotate. So once you select it, you click on the top and you can just rotate it as you like, whatever angle you want to. So it's almost parallel or in the same direction to our field. It has been rotated, but we want to move it and place it on our field, our main experimental field. So we're gonna place it here. So let's just see. And let me just show you here, if you can see between each strips, I have created a buffer zone. So here is buffer zone, here is buffer zone. We want to place our like these straight lines just right at the middle of our buffer zones. So how we gonna do that is by selecting our strips and try to move, hit the move button and try to move it as to the right side of the field a little bit, but let's check where we are with our position. Is still, you can see this is one line of our crops is off of these strips. We want to take it inside and place the line just at the middle of our buffer zone. So we select again and try to move it a little bit to the right. Hopefully it's very, since it's very small plot, you should be very good at it. Otherwise you cannot maybe adjust it according to your like strips. So again, we have to move a little bit to the right. And now it looks nice, I guess. Yep, this row is inside, and here is almost at the middle, but it's a bit to the left, I guess. So what we have to move it a little bit. Okay, let's see. Across our field. Yep. So again, here is a little bit okay. It means we have to rotate a little bit until we are in the same direction, the start and end of our strips. So let's just rotate a little bit to the left, and hopefully this time it should be fine. Okay, it's a little bit, we need to move to the right because it's taking over, it's not at the middle of our buffer zone. Let's go to the downside. What's going on here? So we have to rotate it a little bit to the right because here it's already on the right side, but if you come to the top, our strip lines is to the left, you see? So it means we have to rotate our strips a little bit to the right. So now hopefully it should be fine now. Let's see. Oh, it's a little bit, okay. A little bit to the right. Let's check. Okay. So here is to the left. Oh, okay. So we need to move, rotate a little bit to the left, I guess. And now it hopefully should be fine. Okay, now it should be fine, hopefully. And hopefully it should be fine, but still we need to rotate a little bit to the right. A little bit, okay. Not much, just a little bit, okay. So here is a little bit to the left, here is at the middle. Okay, so sure. Here is, should we rotate it to the right or left? Here is to the left, I guess. Yep. Here is to the left, but here is at the middle. So we need to move a little bit to the left, I guess. Now it should be fine, hopefully. Okay, now it's here is at the middle of our buffer zone. Let's go to the top. Here is a little bit to the left. Okay, so again we have to select and rotate to the right side a little bit. Very okay. Now it should be fine, I guess. Yep, yep. Yeah, now it looks nice, I guess. Yep. So you can see all our strip lines are at the middle of our buffer zone. So it means we are good to go. So what we can do is, so you can see here we have mixed of strips. This is fully grass, covered by grass, our first strip. Second one is half grass, half bare soil. And these 1, 2, 3 are our wheat trials. These are strips. We can call it like replication 1, wheat, replication 2, replication 3, and the last one is bare soil. There is no grass inside the only spot, one or two or few, but it's mostly 90% are soil, bare soil. Now we want to compare and run our statistics, but first of all, let's have a look at our attribute table of our uh, like um, our grids or polygon that we have created consists of ID, left, top, right, bottom, row index, and column index. So you can see our ID, how you can determine which number is one. For example, you click on that, this is our gross. 3 is semi-gross, for example. 3 is our wheat trial replication 1. This is wheat trial replication 2. This is our wheat trial replication 3. And this is our bare soil. So we want to here, for example, using this ID, we cannot understand which treatments are. We want to add a nice name to our field, a nice name to our fields, for example, like nice name to our fields, instead of the ID, we want to have our treatment names, for example, gross, semi-gross, wheat trial replication 1, wheat trial replication 2, wheat trial replication 3, and bare soil. How we can do that? We can just right click and make sure it's the toggle editing mode is on. Otherwise, we will not be able to create another field. So right click on this layer and open attribute tables. This screen will pops up. Here you have ID. What you can do is just let me make it a little bit bigger for screen, for example. You here is a different option you have. This is the toggling editing is on. So here is tools called Open Field Calculator. You can use Control I or just hit this button. You hit once, and here you have represented with a bunch of options, two options. For example, you create a new field or update the existing field. So, but for this, we want to create a new field. And this new field, we call it as a treatment, for example. And since we want to enter our treatment names, those will be text, not number, for example, not integer or anything else. So here we can create an expression for each of our like so each of our so each of our mhm, strips or treatments. So let me create an expression. You can write as a case and then enter. you can, for example, when your ID inside double quotation, you can add as a when our ID, means whenever this is our ID, for example, when our ID equals to 1, okay, then call it as a, then replace it or give the name of the treatment as a gross, for example. Then when our same you repeat for all the IDs, then your ID, for example, equals to the next one is 3. When our ID is 3, then the name should be given as, it should be as semi-grass, for example. And then you hit enter. Again, you do for all the like field ID that you have. When our ID is, for example, 5, the next one is 5. Then inside quotation, single quotation mark, you enter the name that you want to give it to that. For example, this is when our ID is 5. This is our with trial replication 1. And when double quotation ID is, sorry, when our ID is, for example, next one is 7. This is then name it as inside the single quotation, you add the with underscore replication 2, for example, we give it a name. And then then we have our, so okay, let me copy it. Let me just let me just minimize our screen that you guys can see what do I mean by 1. For example, 1 is the gross, 3 is semi-gross, 5 is with trial replication 1, this is 7 is with trial replication 2, with trial replication 3, and this is our bare swell. So that's why let me go to our field calculator. Here I have paste that expression that we have created. So then when inside double quotation, when our ID is, for example, when our ID is, the next one is 9. When our ID is 9, then you, then inside single quotation, you read as a with replication 3. So then the last one is when our inside double quotation ID is ID equals to, this is the last one, 11, which is then it should give a treatment name of bare soil, or just we write it as soil. Okay. Now, once you're done with it, you just hit enter and write down end. So that's it. Once you have, sorry, this was our field that we have created, like treatment. And this is we text start, since the names are in text, like grass, semi-grass, these are in text. So we select our text and create a new field. And once we are done, so we hit the OK button. So now you can see treatment, another field has been created. For example, 11 is soil. That is our 11. 11 is our soil. This is given. 9 is with replication 3. 7 is with replication 2. And 5 is also with replication 1. And 3 is semi-grass. half of them you can see half is grass, half is bare soil. So we call it semi-grass. And the first one is grass. It is fully covered by grass. You can see the whole like strips. So once you have created your treatment name, you give a nice name to them, then you can run your zonal statistics. So next step is we go to processing. You click here and there is a toolbox pops up. So here you can see a processing toolbox pops up a screen. Here you search for a zonal statistics. So here the three options is pop up. So first is raster layer zonal statistic. Next one is zonal histogram. And the next one is zonal statistic. This is the right one. Double click on that zonal statistics. You will be presented. You have to select your input layer. This is our grids that we have created, strips that we have created. We don't want to this side, for example, this side. We only uh, focus on our trials, for example. These are the our strips for our trials. Then in terms of your raster layer, you include your NDVI map, and then you can give any suffixes that you like. NDVI, for example. Here is the statistics to calculate. Click on the three dots, and here is a bunch of statistics are provided. You can select anyone you like, but for this workshop, we only go with the count, sum, mean, and minimum and maximum NDVI. Once you have selected, you can just click OK. And here is you want to save as a permanent file. If you want to, you just click on the three dots, save to file, then it will ask you where do you want to save it and give it a name. So, but we want to create a temporary layer, so we don't need to touch this box. We hit the run, and it takes a bunch of time, and depending on the size of your imagery or image and the trials that you have, the larger your trials are, the larger uh, it will be processed. It takes much longer time to get your data processed, but if you have only small plots like this one, it shouldn't take longer. So let's just hope it's 16%, and 16% is processed. Hopefully it will reach to 100. Now it's 33%. Next, it will hopefully, it won't take long. So now it's 50%. Yep, cool. And the next one will be like, shouldn't take long. Come on, 6%. Yep. Okay, 66%. We're almost there. Okay, now it's complete. Once it's complete, you close this window, and here is your zonal statistics has been displayed. You right click on that layer and come to the export and select the save feature as. Okay, you can save it as a layer or GIS layer project, but here we want to save it as a CSV file, for example. Click here, and here is a format. Different formats of file are provided. You can select and save this statistic result in any format you like. But for our case, we want to go with the CSV file, comma separated values, and here we select our location of this statistics that we want to be saved. Here, for example, we name it as a NDVI underscore, Zonal Statistics. Then OK. So then hit OK. So it should be saved now. Now we go to our our desktop. It was saved as a student. Yep, this is the folder that I have saved it. So you can see here is our NDVI Zonal Statistics. You try to open that file and you can only read it. Select read. So then let me open your GIS also that you guys can easily see the difference. Okay. So now let's go. Here we have a bunch of extra information that we don't need for today. Just try to remove them. And here is our treatment name, grass, semi-grass, with replication 1, with replication 2, with replication 3, and the soil. These were all our strips that we have created here, for example. This is our gross, semi-gross, and so on and so forth. Here is individual count, for example, number of pixels within each strips. They have counted for us, and individual sum, and then individual mean within each strips, and individual minimum within each strips, and individual maximum value for the individual within each strip has been presented here. So, but here we only focus on the mean, individual mean value, and you can come to the conditional formatting. Let me give it like a nice color for you that you can easily see the values. Okay, so let's just go one by one. You can see. Okay, let me open the attribute table and display it nicely one by one. We can hopefully see it the difference. So you can see the NDVI. NDVI actually shows the health of crop or whatever it is on the land is located, land cover or cover the land. It shows the greenness, for example, how healthy the crop looks like. So the first one you can see the gross is has the highest NDVI value. That makes sense because all of the land cover or land soil is covered by the gross. That's why the NDVI value is higher. And the next thing is the next one is you can see this is our gross. This was our gross, so that's why it should have the highest value. And the second one, in terms of ranking, we have semi-gross. Semi-gross is the second strips. You can see half is covered by the gross, half is by the bare soil. That's why the value is the second in terms of ranking. And next one we have with prior replication 1. So with prior replication 1, for example, has been ranked as a third because you can see this one has more width, which are healthier compared to the two other replications. You can see more widths here with these strips compared to the two others. And yep, that was our first trial with replication 1. And the next one is our with trial replication 3. With trial replication 2, with trial replication 3. This is ranked the next because it consists of more weeds and more greener compared to the middle replication 2. So you can see with the bare eye also, with the naked eye also, how the difference. So, and the next one we have is our the next one is 1, 2. Yep, replication 2 is ranked as the fourth one, as a fifth one, and the last one is ranked has the lowest NDVI value in this experiment. For example, which makes sense because our soil strips is consist of only soil. There is only a spot of like vegetation you can see, so that makes sense. So this is called zonal statistics, or for any experiment that you have for each trials. So you go through the same process. It doesn't matter whether it is an NDVI map or any map. You can run these statistics and have interpret the result. So that's all for this workshop. Thank you so much.